On the 25th of April this year, we are celebrating the National DNA Day to commemorate the completion of the Human Genome Project in 2003 and the discovery of the DNA structure in 1953, as well as we are highlighting and celebrating the recent advancement in genomic research and how they are impacting our lives. DNA is a genetic blueprint for life. It's a molecule that carries genetic information for development and functioning of the organism that is inherited from generation to generation. It is a polymer composed of combination of 4 nucleotides made into 2 strands that are wind around each other into a helix shape. And the structure was discovered by James Watson, Francis Crick, and Rosalind Franklin in 1953. The complete set of DNA of an organism is called the genome. One copy of the human genome consists of around 3 billion base pairs. And if you stretch the DNA from one cell end to end, it would be around 2 meters long. And yet the nucleus of the human cell, which contains the DNA, is only 6 micrometers in diameter. So the complex task of packing the DNA into the cell and also its folding within the cell, it's fundamentally important for understanding how the genome functions. The sequence of the 3 billion base pairs that makes up the genome was determined by the Human Genome Project. It was a landmark global scientific project that started in the 1990s, and it was completed in 2003. It was one of the most ambitious and important scientific endeavors in human history. It really kicked off the era of genomics. And indeed, the genetic research has transformed the understanding of human disease and how we diagnose and treat it. But there is still so much more we need to do. Most drugs fail in clinical trials, and there are many diseases where we still have no effective therapies. And now, indeed, we understand what is the sequence of the human genome. However, we are still uncovering how the genome functions and how aberration in this process lead to disease. And it was a surprise to the scientific community after the human genome was sequenced that genes which encodes for protein make only 2% of our DNA. And the remaining DNA was called the dark matter of the human genome in comparison to the dark matter of the universe, as it also makes majority of the genome, and we yet back then did not have the tools to understand what it does. This 98% of DNA was initially also incorrectly called the junk DNA. And it was considered to be a genetic wasteland. Incorrectly, as the dark genome is far from being junk. It has regulatory function. It defines how the genome, the one genome that we have in almost all cells, translates to hundreds of different cell types that build our body. And this dark genome contains regulatory switches that are turning genes on and off at the right time and at the right level to determine cell type specificity. So you can imagine that turning the wrong gene on or producing too much or too little of a protein can lead to a disease. And indeed, 90% of disease-linked genetic changes are located within this dark genome, and majority has no function ascribed. And this includes diseases like rheumatoid arthritis, multiple sclerosis, lupus, heart conditions, metabolic diseases. And this is because those genetic changes can be often located at a large distance from the gene they affect, even 1 million base pairs away. So decoding which genes they dysregulate represented a significant challenge, but also a significant opportunity for drug discovery and development, as is well understood that genetic evidence are doubling the probability of successful approval of drugs to market. So to map the genetic changes to the affected genes, the key is to look on the DNA in 3D rather as a flat sequence of letters and string. And this is because the regulatory switches are coming in close proximity and physically interact with the gene they affect. And we at Nucleome have a unique ability to map those connections at base pair precision. We combine this with machine learning and molecular biology techniques to then confirm that the impact of the genetic change on the affected gene, so to really confirm the causality. And because we're doing all of this at scale, we decoding hundreds of those genetic changes at the same time, mapping the disease-affected pathways at the cell type specific level. This enables us to directly connect genetic alteration in the dark genome to drug targets, specific cell types, potential treatments. We are applying our platform across a range of diseases with high unmet need, with initial focus on autoimmune diseases such as rheumatoid arthritis, multiple sclerosis, lupus. We are building a robust pipeline of novel first-in-class therapeutic assets together with biomarkers to support effective patient stratification. But the technology can be utilized beyond target ID. It can support target validation, patient stratification, but also selection of indication for existing drug with a support of genetic evidence. And we at Nucleome believe that the key to understanding and curing many diseases lies within the dark genome. And we are committed to shine the light on this uncharted territory of the genome through our knowledge and the technologies and turning those insights into precision medicine.